Hi, everyone. Okay, I think it's a good time to start. Welcome back, welcome home to systems biology. We had a long break, holidays. Then I was in Paris last week. It was très bien. I'm back, so we can. I'm glad to see you. I missed you, and the new people are coming in. So let's all together take a nice deep sigh of relief as we start our number 6 lecture. Lecture number 6. So you know we're in lecture number 6 out of a total of 11. The first 4 lectures had to do with network motifs. Let's take another nice deep sigh of relief. Welcome. We're about network motifs, the idea that complicated biological networks are made of repeating occurrences of circuits that you can understand on the back of an envelope and build more complicated networks from those circuits. And then we switched to the principle of robustness. We talked about robustness in bacterial chemotaxis. Robustness, of course, is the principle that says that that biological circuits have a special design that makes their important function insensitive to naturally occurring variations. So this principle helps us, in fact, out of many possible mechanisms for a given function, zero in on ones that can work not only on paper but also inside the cell. And it turns out to have a huge effect in what biological circuits look like. So we saw in bacterial chemotaxis a design that can make the tumbling frequency change after a signal and then return back to its pre-signal level adaptation. We saw how that feature of exact adaptation could be robust with respect to variations in the concentrations of the proteins that make up the chemotaxis circuit. And it's not easy to do that. There's, in fact, just one way to do that with the bacterial chemotaxis on those time scales, which has to do with the principle of integral feedback. So that was what we did last time. And today we'll talk about robustness in a different context, and also a very wonderful biological context, which is the miracle it's really a miracle, even after knowing all the molecules, of how an egg turns into an embryo, into an animal. And this is one of the most amazing things in the world. You start out with a single cell, which is a fertilized egg. And it starts dividing. 2, 4, 8, 16, 32, right? All those cells have the same DNA, same genes. But at a certain stage, the embryo starts to form different kinds of cells. Muscle, bone, skin, brain, heart, right? Despite the fact that all those cells have the same DNA. If the cells all made the same proteins, the embryo would be a blob of cells, all the same, shapeless blob of cells. But the cells start making different proteins, so that some become muscle cells, some become skin cells. And when you think about the patterning, I mean, exactly, it's done with almost no errors, right? You think about how much information is encoded in the shapes of the bones and muscles and skin, the brain, all the organs, etc. Think of this amazing pattern with millions of details that needs to be formed So this is the problem of pattern formation. And the word in biology to describe how you go from an egg to an embryo is called development. Developmental biology is a huge field. So we'll talk about this process of how you form patterns, spatial patterns, shapes. And how you can do that precisely, how you can make the shapes precise, despite the fact that the circuitry that determines the shapes is made out of proteins which are inherently variable. You can't specify how many proteins will be made at a certain point, because one embryo will have 10,000, another one 12,000, another one 8,000. So that's kind of variability. With noisy components, variable components, how can you make robust patterns? Is the question we'll discuss today. So this is the wonder of developmental biology, and the idea that cells have to take on different fates. The fate is a word like to become a muscle cell, skin cell, blood cell, etc. This reminds me when I'm erasing, there are these Buddhist monks that make these amazing sand drawings, very elaborate sand drawings, and then at the end of the day they erase it. And this to symbolize the fact that things are created and then die, right? So this is very important to remember. Okay. So how to make precise patterns with noisy variable protein circuitry. It's a question of robustness. So let's start with how patterns are formed at all. This requires positional information. Cells need to know where they are in respect to the rest of the body in order to make this decision to become their appropriate fate, to assume their identity. And the way this works in many systems is by means positional information is conveyed by special shape-making molecules called morphogens, which how many people here have heard of morphogens? Okay, so about half the class. It's a very well-known concept in biology. These morphogens are produced. So imagine this is a piece of tissue which we want to pattern. This is called the field of cells which are initially identical. We want to make a pattern here. Morphogen is usually made at some source of morphogen, which we'll call this M, and it starts diffusing. And usually it also gets degraded. So it makes a concentration profile which is high at the source and then lower and lower into the field. This is the position. This is the morphogen concentration. Of course, you can ask, wait. So already has some information. The morphogen is made at a certain point, right? So it's a chicken and egg problem. Where does the original information come from? And a lot of times that comes from, for example, the mother, which already has a pattern, the chicken, deposits something at one end of the egg, and that's the original source of information. And then later on, once you already make a pattern, each pattern can make morphogens for the next finer pattern. So it's a sequential process of finer and finer. the way this works, I just want to say the prototypical understanding of the way this works is called the French flag model. Which was introduced by Wolpert in the year that I was born, 1969. And it's the following. It's called the French flag model. It says, here's the morphogen diffusing, right, and degrading. So it has a concentration profile. And the cells here have receptors, just like in chemotaxis. Remember, the receptors, the molecules can bind things. They have special receptors that can bind the morphogen molecules. So the cells can tell what's the local concentration because they have receptors that bind the morphogen. And there are thresholds of concentrations. This is one threshold. This is the second threshold. And cells that are where the concentration is lower than this first threshold, they decide, oh, I'm probably close to the source. So I'm going to become, let's imagine, cells of the head. And cells that are lower than T2 detect, they say, oh, I'm probably far away from the source. I'm going to become the tail. And cells in the middle say, I'm going to become the belly. So in this, if you have two thresholds, you end up with three cell types. You patterned the field into three classes of cells, A, B, and C. Right? So it's called the French flag model because, you know, if red, white, and blue, right? French flag is like these types like this, as I know very well from my visit to Paris, where you see a lot of French flags. So this is the French flag model. It's a conceptual model. Okay? So, now, experiments, I would say for a long time, development was, you know, of course, one of the miracles of biology for almost in all our history. Was it's a miracle that you can't explain. And in this last century, this century, experiments tried to ask things like, if we change something about the cells, like temperature, if we add all kind of chemicals, is it easy to perturb the shape of the embryo, to ruin the patterns? And overall, I mean, not in every system, but in general, the pattern. Actually, Owen, I want to ask you to bring a better black one, because I can't. This is going to run out. The patterns are robust to perturbations. Perturbations. For example, there's a classical way of experience where you change the amount of morphogen that you have. For example, by deleting, mutating one copy of this morphogen gene that produces the that encodes the morphogen on one of the two sister chromosomes, because you know that animals have two copies of every gene. So you knock out one gene, and you can see that the morphogen concentration goes down by 50%. There's no correcting mechanism for this. But a 50% reduction in morphogen concentration, usually the pattern remains unchanged. Okay, so, for example, morphogen source, you multiply it by 1/2, the pattern is virtually unchanged. And this kind of phenomenon was called in the 50s canalization by a very important biologist called Waddington. Canalization, which means that if this embryo development, once it starts, it goes into like a channel, like a canal, that guides it to the end. And this canal, it's very difficult to get. It's like a roll. He had a picture of a rolling ball down a cliff, which has a canal in it, and goes into the canal. You can't change. So this kind of idea. But it wasn't until this decade that we know about mechanisms that can actually explain how this happens. And I'll just say intuitively, you know, if I drop the source to 50%, Okay? You know, and I keep these thresholds the same. What happened to the pattern? A, what happens to area A? It disappears. And what happens to area C? Instead of yeah, it starts. So it's a pattern that looks like B, C, something like this. It's an embryo that has no head and a big tail. Okay, so that's what you expect to happen, just naively. Okay, so, but it doesn't. It doesn't. You drop this to 50% and you get the same pattern. So how can that be? Huh? Okay, so let's start analyzing it and see.
Or they change according to the maximum.
Yeah, so the thresholds don't change. The thresholds are kind of encoded in the receptors and in the genes. Thresholds don't change. So that's the wonder. All right, so that's our system, diffusing, degrading morphogens, and now we start to analyze it. So here we notice it's robust phenomenologically. So now we want to start making a model of this and to understand its the way it behaves. I should ask if there's any questions. I don't mean questions predicting the answer, but questions about the details here. Something I didn't explain myself. Okay.
There is something called allelic exclusion. Usually one gene of the two alleles expresses itself. So if you delete one, probably the other one then gets expressed. That's why there is a compensation.
So Lydia asks, if you delete one copy of two genes, does the other one know about that deletion and increases in concentration? So in these experiments, you can measure what happens to the morphogen. And in these experiments, you delete one copy of the two genes, and in fact, the concentration of the morphogen at the source goes down. There's no compensation. And that's quite general. There's an experiment now came out, Springer lab, in yeast. They looked at deleting one of the two copies of every gene in yeast and putting green fluorescent protein on the other. And in general, the gene that's left doesn't know that there was one gene deleted. There's not a lot of compensation. Let's say 80% level in a given condition. So that's interesting. So I want to just write some simple model so we can start working with it. And this is diffusion plus regular degradation. I want to show you that this speaks to an exponential morphogen gradient. And this is not robust. So I'm translating into math just a little picture that I drew for you. So our morphogen concentration, this is its rate of change in time. The math of diffusion has to do with the diffusion equation. How many of you know what the diffusion equation is? Okay, so about, again, half of the class. And the rest of you, I'll just give a very quick explanation. And this could be a good subject for the exercise class. Diffusion has to do with the second derivative of the concentration with respect to position. By the way, I'm doing a one-dimensional model. Why? Of course, because it's easy to solve, but also in real life, a lot of these processes, of course, our patterns are three-dimensional, but the biology is divided into one kind of quasi-one-dimensional problems. There's a morphogen for head-tail, the morphogen for front-back. There's, once you make these patterns, inside our segments, there's morphogens for this direction, morphogens for this direction, morphogens for this direction. So they're broken down. Sometimes there's a kind of radial symmetry, like in plants. You have the petals, etc. Sometimes you need the intersection of two different morphogen gradients to make patterns. So there's a sequence in time, and it's broken down into quasi-one-dimensional problems. And there's additional processes. So morphogen is one major way, but there's additional things that happen, especially in organisms like us. Cells also move from place to place. There's growth. So during growth, things happen. Cells die. So for example, there's a blob of cells, and then these part between the fingers, all these cells die in a programmed way to make the space between the fingers. Cell death, adhesion, a lot of additional things. So this is just one principle. There's a lot to discover in this field. It's really wide open. Many more questions. How the brain is patterned, really a mystery, etc. So this is D, is the famous diffusion constant. And this term, the second derivative, is a nice thing. It tends to, if your concentration profile looks like this, here it looks like a parabola. This parabola looks like something like minus x squared plus bx plus c. If you take the second derivative, you get something negative. So parabolas like this go down. If you have a place with no morphogen, it looks like a parabola here, ax squared plus bx plus c. Take the second derivative, you get a. So this is positive. So if you have a shape like this, diffusion wants to make it go down. If you have a shape like this, it wants to fill it up. Diffusion wants to smooth. That's this term, second derivative. And there's degradation. So this is degradation. Right? Very simple. The rate of change of morphogen is the diffusion, how much it comes in from the sides. And it gets degraded. All these morphogens, or a lot of them, get degraded. Of course, a key equation in physics, heat. We also have boundary conditions. So our problem, this is x. We have our source here, which has a concentration which we'll call m0. M at x equals 0 is m0. It's the source concentration. We assume that the source concentration is fixed. It's the amount of morphogen produced. It's more natural maybe to take the flux to be fixed. You can also solve that, but for simplicity, I'm taking the concentration will be fixed. And the other boundary condition, it's a second order equation, so we need another boundary condition, is that m at infinity, far away, it's a big field, is 0, which means to say that m is going to decay away. In infinity, there's no more. So we need to solve this equation. The answer will be that m is going to show us an exponentially decreasing profile. And then we'll calculate the thresholds, we'll do the French flag model, we'll see what happens when you change m0. That's our program. All right, so I'm going to solve this at steady state. That's to say, I'm going to assume the system has enough time to diffuse and be degraded, until there's some balance between these two processes. So steady state, of course, means that there's no more change. You have 0. This is 0. And so the equation that we need to solve, no more, right? This is 0. So we need to solve what function, when you differentiate it twice, is equal to itself. Huh? Sinus? Sinus is actually equal to minus itself. Good. Exponential. So we're going to guess an exponential function. This is the Greek letter lambda, which is the typical scale over which x decays. We're going to guess this function. And now we're going to, we're going to differentiate it twice according to x. So if we differentiate it once, we get minus a over lambda, x over lambda. If we differentiate it twice, another minus 1 over lambda comes here. So it's a over lambda square e to the minus x over lambda. That's fine, right? And this equation says that this second derivative which is with d a over lambda squared e to the minus x over lambda equals alpha times m, which equals alpha times whatever it is that I guessed, a e to the minus x over lambda. So I plug this here, I differentiate it twice, alpha m. And now I can calculate what A and lambda are. So first of all, of course, conveniently, this fall is, and we see that A also goes away. Fantastic. And so we have that d over lambda squared equals alpha. Or in other words, lambda equals square root d over alpha. So this is the typical, so what we get here is an exponential, I'm going to draw it here, this is m0, we get, yeah? This one? So what I'm writing here is, see, because d second derivative equals minus alpha m equals 0, then I move this here, right? So I get that this equals that, right? Is that clear? Okay. Therefore, I equate the second derivative to alpha times m. Did I explain myself? I cross this, I cross out everything that's... Thanks for asking. So this typical scale, lambda, is the scale at which the Morphogen gradient goes down to 1 over e, to 30, 1/3 of itself. So this is the typical scale here is lambda. That's the typical size of the patterns. And you see that the faster the diffusion, d, bigger d, the bigger the patterns, the smaller the degradation, the Morphogen can go farther, right? So this is actually the length over which the Morphogen can diffuse before it's degraded. That's the typical length. And what's A? We can get that from this boundary condition. M at x equals 0 is A e to the minus x equals 0. So A equals m0. So our solution is that the Morphogen is m0 e to the minus x over lambda, and lambda is that. That's the way you solve the diffusion equation. If you're lucky enough to be able to solve it, sometimes it's more difficult. Now let's ask, is this robust with respect to changes in the source? So what we're going to do now is change the source to something else and calculate the shift in the profile. So what I'm going to do, I'm going to change the source, let's say, to a lower amount. We know what the profile will be. It'll just be that lower amount times the same thing, right? Because we didn't change the diffusion constant, we didn't change alpha, we just changed the source. So we have an exponential, the same typical length, except it's starting at a lower level, at x equals 0. So what does that look like? Looks like this. And there's a shift . There's a shift to the left of the concentration profile. And as a result, there's a shift to the left in the entire pattern. Because the pattern cares when you cross those thresholds. The pattern is based on sensing the concentration at your own position. If the whole concentration profile is shifting to the left by a certain amount, the entire pattern is going to shift to the left by the same amount. So, if I had a threshold, here, then the point where you cross this threshold shifts to the left by delta. So let's calculate it. So let's calculate. Let's really put here, this is the threshold t. This is the point x 0, that this is where you cross the threshold. This is, let's say, when you go from state A to state B. So to calculate this position, I ask, where does the concentration equal this t? And then I need to find what this x0 is. And the answer, we did this kind of calculation many times. E to the minus x0 over lambda is t over m0. x0, I need to take a logarithm, minus lambda log t over m0. If I put in m0 prime, I get x0 prime. x0 prime is the same thing, except I have to put in m0 prime here. So this is the lower concentration. And the shift, which is x0 minus x0 prime, is minus lambda log t over m0 minus lambda plus lambda log t over m0 prime. And the difference, the lambda is the same. The difference between two logs is the ratio of what's written inside. Delta is just to understand how much this is. If we take half.
Oh, it's without the minus.
What? Without the minus here? If m0 prime is just 1/2 m0, that's to say, if we lower the concentration by 50%, right? We lower the concentration by 50%, then this shift is lambda log Stein, which is 0.7 lambda. So if I lower the concentration by a factor of 2, the whole pattern shifts to the left by almost an entire lambda. That's to say, by this 0.7 times the typical size. It's a huge shift. It's a shift. It's a very large shift. It's on the order of the entire pattern size, and therefore we have no robustness here. Robustness to changing the initial concentration. All this calculation is just to show that the simplest interpretation of the French flag model, degradation, diffusion, doesn't explain the robustness you see in the experiments. On the other hand, it's such a generic model. I mean, what can you do to make things more robust? What are we doing? So one answer was given by the first mechanism for robustness, molecular mechanism for robustness, and then several others here at Weizmann by Nama Barkai and our graduate student Avigdor Eldar, who's now has a lab at Tel Aviv. And they took a look at this problem and decided to start from the same degradation and diffusion, but to make a more general model where you can have some more mechanisms for robustness. And what they did was to take a look what happens when you you use the same diffusion. So what is here is a mechanism for robust patterning. But instead of having a linear degradation term, they now allow a more general function, like nonlinear degradation. So. By the way, there's very few things you can actually play with this equation, because when you think about, you can't, for instance, make the parameters depend on space, because the field of cells initially is unpatterned. So the morphogen can't, the field itself things there can't depend on the position before you pattern them. So you have to play with somehow with the shape of the equations, not with their parameters. They can't depend. And they noticed that this equation has a very nice feature, which is called translation symmetry. That's to say that if m of x solves it, so does m of x plus delta. If you make a uniform shift in space, you still have a solution. Could be for different boundary conditions. You still have a solution. And the reason is because if you change this to x plus delta, the derivative doesn't care about it, and everything here doesn't depend on space. So because of this property, suppose you have a profile, something that solves this equation. And I start with a different initial condition. Then if you take this profile and you shift it to the left, so that it hits this initial condition, this shift is always uniform in space, you also get a solution to the same equation. That's to say, in this kind of equations, if we have a solution and now we change the source, to find what happens, all we need to do is to take the solution and shift it until it intersects this source position. And this shifted line is the solution to this equation. So I'll show you mathematically this. And that means we're in trouble, because if we lower m0 by a lot, we'll get a big shift. But there's one way to get around it. And that is, if you want to get small shifts, make this equation so that you drop quickly here. Because then, to understand what happens if I reduce the source, the shift will be very small. So make m drop rapidly, steeply, near x equals 0 to get small shifts. So how can you do that? Well, with our previous exponential gradient, if we make, in order to get a steep drop, we need to make this very small, right? So that the exponential drops near the source. But that means the patterns we can make are very small. So we need for robustness, one, steep drop near x equals 0. And two, we need long range, because we want large, we need to make a nice big pattern. So how can you get this? They looked at this and they found a solution that turns out to explain the detailed structure of a lot of developmental circuits that before that didn't have a good explanation. And in fact, I'm going to tell you what it is after the break. So let's take a 10-minute break and come back at 3:05. כן, אני רוצה להתחיל. Okay, let's start. יכול לקרוא להם? כן, אני אסיים בנושא הזה, אני לא הולך עכשיו לבנות על זה. אני דילמה. אפשר לגמור היום לפני הזמן פשוט. זה די מסובך, לא? All right, let's start. Okay, welcome back. If you feel like it, I invite you to take a nice deep sigh of relief together with me. So we looked at the diffusion equation. We saw that it gives exponential gradients, not robust to shifts in the initial conditions. And now we tried this nonlinear equation. We talked about translational symmetry. And it turns out in order to get robustness, you can do it rather easily. What you do is you make degradation is self-enhanced. So what's going on here? Instead of regular degradation, which is linear in the concentration, which means each morphogen has a certain clock, or let's say, as a typical time afterwards it's degraded. In this case, the higher the concentration, it goes like m squared, the higher the concentration, the bigger the chance for degradation. I mean, to remind you, before the break, we saw that you need to go steeply here and then have a long range. So that if we make a shift, we hardly have an effect. So this could work, because where the concentration is high, degradation is fast. When the concentration is low, m squared, degradation is very low. So if you manage to make it away from the source, you can diffuse a long way. You know, you might have said, okay, let's make the degradation constant fast here and then slow here. But can we do that? We're not allowed to, because the field of cells has no pattern in it. We can't make the degradation rate depend on position before we have positional information. So we can't do that. Can't do that. We have to work only with the properties of the molecules. And here we make degradation self-enhanced. Anything that, where there's more morphogen, the faster it's degraded. And we'll model it here as m squared, this kind of term. But in fact, anything that makes the degradation faster when there's more morphogen at that position. Any questions?
Question about the assumption of diffusion. You have a tissue here with membranes between the cells, so why can you assume that the morphogen can diffuse freely through the membranes?
Thank you. So the question is about diffusion and how can it diffuse through the membranes of the cells. So there is something I didn't explain. These are the cells in the field. The morphogen is above the cell. It's usually in a layer of water touching the cells. It's not inside the cells. It doesn't go from cell to cell. It goes on top of the cells. And then the cells have receptors that can sense the morphogen. So that's usually the way it happens. There are some morphogens that depend on cell-cell contact, notch, for example, but that's a different story. But these kind of morphogens I'm talking about are diffusing over the cells. It's a really good question. Other questions? There was one question in the break. Great, if you make this means robustness. If you make the morphogens smaller, the pattern doesn't change. So how can we have dinosaurs and small lizards, which have the same, like say, very similar pattern, but such a huge size difference? That's a different problem. It's called scaling, and I think only in 2008, Nama Bakai and Danny Ben-Zvi published a paper that offers the first solution to this problem. It was unknown before that how that works. Maybe I'll teach that next time, next class. We'll see. Other questions? Yeah.
If the signal M is noisy, then the thresholds have to be very far from each other, right? But you have a long tail, so how can you not have from like A, B, A, A, B?
So the question is that if this, so this is a threshold, and this determines the position of let's say one region. There's another threshold position of another region. So if the morphogen is noisy, so in fact looks like this, you might cross this twice. That's a great question. I mean, I don't have a ready answer for that. Maybe an open question even. Other mechanisms to correct for that. I would say that the M usually makes a kind of a first order separation into patterns, and then there are correction mechanisms. That has to do with, for instance, cell-cell contact and movement and things like that. But in general, the diffusion is quite smooth. It's smoother than, because of the smoothing properties of diffusion, you don't get a lot of this. Usually in diffusion processes, you do get very smooth, like not as noisy. But so let's try to solve this equation now. And with our what we did in the first class, this is going to be not so hard, actually. And we'll see what the profile looks like. So again, we're going to do steady state. Steady state means no change. And that means that it's a function. Which function is it, I'm going to ask? When you take the second derivative of that function, it's equal to itself squared. Right? What kind of function, when you take its second derivative, is equal to itself squared? See?
1 over x squared?
Yeah, so it's like some kind of 1 over x something like that, exactly. So we're going to guess, I'm going to just guess the right solution. That's a lot of times the way we solve differential equations. We know the answer. And I'm going to guess a polynomial, something to the power n. We're going to calculate a, epsilon, and n by plugging them into this equation. So let's do that. So dm dx is, you remember when you differentiate a polynomial, you have n minus 1, the n goes here. And when you do it again, the n minus 1 goes here. And here you have n minus 2. So d times this needs to equal alpha m squared. That's what the steady state means. D times the second derivative equals alpha times m squared. So let's plug that in. That's alpha a squared x plus epsilon to the power 2n, because a, that's the square. So let's first calculate n. You see, here we have x plus epsilon to the power n minus 2, and here to the power 2n. So what n is it that n minus 2 equals 2n? Minus 2. So minus 4 equals minus 4, right? So we know that our solution is x plus epsilon to the power minus 2. Now let's, so now if we put in minus 2, this is minus 4, we can erase this. Now let's look at A. Alpha A squared equals dA times minus 2 times minus 3, right? Because I plugged in n equals minus 2, which is 6 dA. Therefore, A equals 6d over alpha. And epsilon, we calculate from knowing that m at x equals 0 is just our initial condition. And that's equal to A 0 plus epsilon to the power minus 2. So epsilon equals m0 divided by A to the power minus 1/2, or 1 over square root m0. Okay, so this is, we solved the form of this graph. And now let's look at it, how robust it is. And the idea is the following. If m0 is very large, then this epsilon, which is like 1 over square root m0, is tiny. So this epsilon is very small. And except for a very small region here, which is about epsilon, except for x's that are very small, so x is very small, we can't neglect epsilon. But when x is big compared to epsilon, we can neglect epsilon. Our Morphogen gradient looks like some constant divided by x squared, which does not depend on m0 at all. X larger than epsilon, right? So except in this small region, which we can make as small as we want, because this size of epsilon goes down. The more morphogen we have, the smaller it is. So we can make this a lot of morphogen. So this region becomes as small as we want. Except for in this region, our function goes down like something over x squared. So this is very different from what we solved in the first half hour. In the first half hour, if you remember, we had the morphogen that looks like m0 into the minus x over lambda, which depends very much on the initial level. You change the initial level by 2, you change it. Here, it doesn't depend at all on the initial level. If you're far away from the source, far enough from the source. Which means we have robustness of the profile m of x with respect to the morphogen source m0. And therefore, in these kind of patterns, we gain long range, because it's 1 over x squared. Actually has longer range than the exponential. It has a very long range function. Exponential, if you go down 1 lambda, you go 2 lambdas, 3 lambdas. After 3 lambdas, you're down to a few percent. After 10 lambdas, you're down to 10 to the minus 5. You can't get more than a few lambdas. With 1 over x squared, you can make very long range patterns with your threshold. So it's a power law. It's not an exponential. And second, it doesn't depend at all on the initial source, except for a small region that you can make as small as you want by increasing the source. And therefore, this kind of mechanism, self-enhanced degradation, gives you long-range patterns that are robust. It doesn't have to be m squared. It can be anything that makes the degradation depend on the morphogen in any way. Questions?
Can you explain biologically what this means, that the degradation is dependent on the...
So the question, can I explain it biologically? I'll do that in the next thing I'll do is I'll show some examples how that works. Yeah. Other questions?
But you're not dependent on X, and that again continuity.
So X is the position, right?
What happens if in the last division you can get 10 cells across the field, you can get maybe 20 across the field, or that?
I mean, the total size of the field. So this also doesn't depend on the size of the field. That would be like the boundary conditions on this side, which doesn't affect this. And this is actually a problem for a model like this. This kind of thing works, in fact, when there's not a lot of variability in the size. If there's no small babies and big babies of the same species. Like in flies, it's kind of fixed. And if you do need to depend on the size of the tissue, then you need a different mechanism that I might do in the next class, which has scaling. It's an additional feature. This doesn't depend on the size of the field. That's right. So you need that to be fixed. Very good questions. Oh, by the way, you can say it does depend on the diffusion constant and on the degradation rate, but those are different. They're not like concentrations of proteins. Diffusion constant is very fixed. It doesn't depend too much on temperature and things like that. This is also the rate, the specific rate of the protease is also like a hardwired molecular number. So you do, if you change this, you do change the profile. Right? Also, if you change the thresholds, let's say make mutations in the receptor, let's say, you can also change the pattern. So you can evolve the pattern. You can make a bigger head, smaller, but you need to make mutations that change the threshold. Right? So,
since this was also dependent, I mean, you said it's robust except for this epsilon,
yeah.
right? Which is also dependent on M0.
Yeah.
So if M0 goes down by half, then, I mean, for example, when you scan mutants, you should see more changes in the vicinity of the source than.
Good. So the question is, if you have mutations that change M0, let's say reduce M0, then this area epsilon where there is an effect of M0 grows bigger. So you might expect mutants where you have very low levels of morphogen, and then near the sources there should be more changes than far away from the source. That's a very good prediction. Yeah. And in general, if you take this source to 0, then there's no pattern at all, right? It can't be so robustness here has limits. So it works. You make M0 so big that its natural fluctuations make this even if it's the lowest you expected with natural fluctuations, this epsilon is small compared to the pattern. That's what you need to. But if by mutations you make it even lower, you'll start to see a range where there's distortions. Yeah, and if you destroy M0 completely, you'll have. So that's important, very important point. That, every robustness mechanism has limits. There's limits where you take the parameters to extreme values, and then it's no longer robust. Otherwise, even if M0 was 0, we would still get a pattern, which doesn't make sense.
How do you compare? Is there any fine measurement which? Is there any measurement which distribution is more robust? Something line, or you only?
So the question is, how do you compare two models or two measurements? Yeah, of course. There are experiments like Nama and Benny Shilo here works on flies, right? Did experiments together. What you do is basically you look at a region. Let's say here's the head and here's the stomach. You look at that position, and you start changing the morphogen level, and you measure how much this shifts. And the ratio, let's say, if you do a twofold change here and you get a 5% change here, the ratio between those twofold changes is a sensitivity. You can say. So you look for the sensitivity is a measure for robustness. The smaller the sensitivity, the bigger the robustness.
Okay.
In general, you take the sensitivity is you take the relative change in x as divided by the relative change in y. That's the sensitivity of x with respect to y. Which is equal also to d log x over d log y. And that's in that way you can quantify the sensitivity of any parameter of anything with respect to anything, and then robustness means that this is a very small number. And in fact, you can get it as small as you want in models like this, and you're looking for models where you can make it as small as you want with a big range of parameters.
This model assumes that it's a lot more robust to assign the detection level. That is, the detection levels are fixed. Can you say a word about why is it?
Why detection level fixed? Thank you. So let's look at this mechanism. Here is the source, which makes these morphogen molecules, and they move, and they get fewer and fewer in number, right? Because of degradation. And the way they're sensed is by special receptors on the cell surface that can bind specifically this morphogen. So if we make the receptors, they have a certain chemical, their shape is such that they have a certain chemical affinity to the morphogen molecule. And who asked the question? Okay, sorry. And that's something you can fix. If let's say if you now move from one kind of fly to another kind of fly over evolution, you can have mutations in that receptor, and those can change the threshold. But now let's just talk about one species of fly. You come with this receptor, so the threshold, which is based on the chemical affinity between morphogen and the receptor, that'll be the same for all the embryos. But what won't be the same for all the embryos? It could be something like the amount of this morphogen. Why? Because let's say one is the mother was starved, and all the egg has fewer yolk proteins, and now all the cells are making less stuff. So this morphogen, instead of having the maximum level, has, let's say, 80% because of this. That happens, right, in nature. But you still want the pattern to be the same. So what I'm trying to say is that the naturally occurring variations in this kind of system aren't in the threshold, but rather in protein levels, which we think is often the case.
You need to explain why, if all proteins, for example, that's also the scaling, I'd also expect the number of receptors to change, so signals might change, so it's not that.
So you say inside the cells, we also need some robustness to protein levels.
No, I'm saying if you produce, for example, less morphogen, you could also argue maybe you produce less receptors.
Yes.
Which will also cause.
Yes. So let's say I'm moving the problem here. So the question is how to make also the signaling systems and the transcription circuits robust, right? So that's like it's again a very good question. That should relate to what I said last time about the bacterial chemotaxis circuit, and there's also robustness should be at all levels, also here, absolutely. So when you look at this signaling circuit, now you say, okay, let's make this robust also to receptor levels and all that, and it is also. So if we had, if the circuit is certainly not robust to changes in the if you change the threshold levels, you change the pattern, unless somehow they change in a correlated way with the profile, right? So you can think of again. I want to say this field is really at the beginning. This paper I told you about is 2004, and the scaling problem in 2008, and that's just like the first examples out of. So there's a lot of work to do here. Good. So I say, so there's developmental circuits. I have network motifs that can provide self-enhanced degradation. So this relates to the question, how do you mechanistically achieve this phenomenon where the more morphogen there is, the more quickly it's removed. And so I'll just give you two recurring motifs. Again, they occur there's probably dozens of circuits that have been worked out in different organisms. So this is the morphogen. It binds to the receptor. It gets degraded. The receptor, by binding to the receptor, that's actually the mechanism of degradation in these systems. Because what happens is that it binds the receptor and sticks to the receptor, and then the receptor is internalized by the cells and degraded inside the cells together with the morphogen. See what I mean? So this is what's called endocytosis. Is the biological word for this process of taking the receptor together with its morphogen.
But by then, it has already already sensed.
I forgot what I wanted to say is the morphogen binding increases the level of receptor. So I missed a step here. Morphogen binding to the cells makes signaling, transcription of more receptors. Bind more morphogen and take it away. Right? So this is so. And then when you reach steady state, the upshot is that places that have more morphogen, it's not a linear degradation, it's nonlinear degradation. The more morphogen there is, the more its removal rate is larger. Hope I explained myself. And the example for this is M equals hedgehog, and R equals patched. These are two names of mutants. The mutants in development have funny names. They tell you what happens to the fly when you mutate. The fly it looks like a hedgehog, like a kipod. Or, etc. So these names are. This is an example, but many other examples. And there's another motif which looks like this. So what's going on here? Here, binding of morphogen to the receptor causes signaling that makes the cell make less receptor. Okay, so M signaling inhibits receptor production. And the receptor, but the receptor interacts with the morphogen to make it degraded by means of extracellular proteases. So this is another. It's like instead of a double positive loop, this is a double negative loop. And the examples are here. So imagine, this is called wingless. You can imagine what happens to flies when you mutate the morphogen wingless. And R is called frizzled. Wingless and frizzled. That's, again, just an example. They don't exactly give you M squared terms, but they give you self-enhanced degradation. Yeah. So the question is, does this threshold depend on the number of receptors? Yeah, in these kind of mechanisms, you want the threshold not to depend on the number of receptors. Okay? So you need to consider what's going on in the cell. So in the cell, there's receptors, and they, as you remember, they phosphorylate proteins. They go into the nucleus. They activate genes, etc. There's all these circuitry inside. And it'll be the job of this circuitry to make robustness in its own domain. Mhm. Yeah.
It could also be a situation when there's two kinds of receptors that bind, and one will be not in charge of the degradation circuit, and one will be the signal. And then that will happen to the
So the question here is, I think, is a dialogue to your question, was that there will be two receptors, one that does endocytosis and another one that does signaling.
And then when you use it, there are receptors.
Yeah. Mm-hmm. I mean, in fact, in cells, in mammals and vertebrates, most receptors, when they bind their signal, do endocytosis, and that's like a very general mechanism to see. But I don't know about models that address this. How can thresholds now be robust to receptor numbers? Maybe they exist. I just don't know about them. So we're touching questions that are at the frontier. You see how close the frontier is? So yeah.
In the first model, are increases from the receptor level, are they transcriptional increases, or is the protein already there?
This one?
Yeah.
Yeah. So this you start with some receptors, and binding of morphogen to receptor causes signaling that ends up expressing many genes, including the receptor.
So isn't the timescale of diffusion much faster than the timescale of transcription?
Okay, so the question about timescales. Let's think about timescales. The timescales of this whole cell-fate determination process, what we're interested in is the timescale when the cells make their decision. So the diffusion is fast. It binds the receptors. There's more receptors made, which affects the concentration of morphogen, etc. But we're looking for the time when the cell says, okay, I'm going to be a brain cell. I'm going to be a nerve cell. And that time can be on the order of a cell generation time, let's say, depends on the system. So the cell cycle time, and that's slower than all these production rates and diffusion rates. So there's enough time for the system. Not always. I mean, sometimes the systems will be out of steady state. That's another kind of theory, because all we did here was steady state, right? But it's not out of the question that this thing happens faster than, so there's enough cycles here for this to reach steady state, and then a cell cycle decision. But
I guess what I'm asking is the endocytosis enough to, by itself, without the increase in the level of R, is that enough to give assisted
so self-enhanced degradation. Yeah. So if we don't have this arrow, then the morphogen destruction rate will depend on the concentration of receptor, and that's constant throughout the field. And so the morphogen the destruction rate won't be dependent on its own. Actually, the more morphogen there is, it'll get rid of more and more receptors, and it'll actually be lower. So that won't work. So in order to make it self-enhanced degradation, we need something like this. Otherwise, it's self-inhibited degradation. And I think instead of robustness, you get hypersensitivity to changes in the initial level. But again, I'm just improvising. It's worth analyzing. Good. You are taking me, you're making me work. It's great. Any more brilliant questions? Or even not brilliant, doesn't matter. Just questions. Don't want to put any pressure. Okay, so we looked at the problem of development. We just scratched the surface, okay, just for you to understand that there's morphogens, this geometrical problem, diffusion, degradation. Within diffusion degradation in physics, you're used to exponential solutions. here biology can show, and this has actually been measured, power law solutions, robust to initial conditions, very non-generic solutions to reaction diffusion equations, and that you can, there's a variety of ways to, so it's like a general principle from simple models, self-enhanced degradation. There's a variety of detailed ways with their own details that we need to work out, just like your questions, right? You can get self-enhanced degradation. And there's now there's a few ways known, shuttling mechanisms, to get robust patterning, which all, for some reason, give power law solutions. This is a 1 over x squared, not e to the minus x. Interesting, not understood why in general it's power law. And one of those fields where this is going to be full of additional principles in the coming decade. I think very, very exciting area of research. And so there's nothing left but to take one final nice deep sigh of relief.